luminous minds gathered here at the International Conference on Microbiology and Microbial Technology 2025, organized by the Faculty of Technology, Rajarata University of Sri Lanka, together with Qujing Normal University, People's Republic of China.
Today, we are celebrating silence, collaboration and innovation all under one roof.
Exactly. So let's keep the energy flowing as we officially begin this exciting day of learning and discovery.
Let's start this remarkable morning with a symbol of light and knowledge, the lighting of the lamp.
We warmly invite our distinguished guest to step forward and light the lamp of wisdom and inspiration.
Dr. Janaka Pushpakumara, Acting Vice Chancellor, Rajarata University of Sri Lanka.
Professor J.D. Bar, King Saud University, Saudi Arabia.
Professor Turki Daud, King Saud University, Saudi Arabia.
Professor Janaka Gunaratne, Dean, Faculty of Agriculture.
Dr. Manoj Fernando, Dean, Faculty of Applied Sciences.
Professor Bandala Jayatilaka, Dean, Faculty of Management Studies.
Professor E.M.S. Ekanayaka, Dean, Faculty of Social Sciences and Humanities.
Professor Chaturanga Bamanarachike, Dean, Faculty of Technology.
Ms. Samanthi Herath, Registrar.
Mrs. Asoka Sirivardhana, Librarian.
Mr. Sampath Godukumbura, Bursa.
Professor Nalini and Vijayavardhana, Co-Chair of the ICCMMT 2025.
And all the head of the departments of Faculty of Technology. Thank you everyone for igniting this event. And ladies and gentlemen, let's rise together for the University Anthem.
Thank you, everyone. Please be seated. It's my great honor to invite Professor Chaturanga Bamnanachige, Chair, ICCMMT 2025, and Dean of the Faculty of Technology, Rajarata University of Sri Lanka, to deliver the welcome address.
A very good morning to all of you. To our friends from abroad, welcome to Sri Lanka. Hopefully you have had a wonderful time up to now. Acting Vice Chancellor, Dr. Janaka Pushpakumara, who has always been very supportive of ICCMMT 2025. All the deans of the faculties, my friends, my colleagues, I sent them the invitation on Saturday and they are here. So this is the kind of collaborations we have with other faculties. Thank you very much for turning up. I know they have been little cross with me for not letting them know the dress code. I'm the only one with the blazer, but anyway, I'm really happy that you're here. Welcome to ICCMMT 2025. Registrar, who has been very helpful arranging the logistics, Ms. Samanthi Herat, thank you for your time here. I know a couple of interviews were scheduled to be held at 9:30, and that has been now postponed because of our event. Thank you very much for arranging that. Then all the foreign delegates who have come to join with us in this momentous occasion, which is the first international conference on microbiology and microbial technology, hoping that this will be an annual feature as we have planned. So this is the pioneering conference, and all the academics and the students of Rajarata University and the participants and presenters and keynote speakers. I welcome all of you to the Faculty of Technology, Rajarata University of Sri Lanka, and especially to ICCMMT 2025. So this was first initiated in January and March when Professor Nalin came up with the idea of bringing together all our collaborators, all our research partners to a single platform where we can share our knowledge and inspire young minds to do even better. And he was instrumental in single-handedly coming up with all the invitations to researchers abroad and collecting abstracts. So that was a one-man show to a certain extent. And then the organizing committee joined in. The last few weeks have been really hectic but very fruitful. The team of organizers have done an amazing job. There were people who were collaborating from abroad as well, pointing out errors in the agenda, and the agenda was a continuing document. But however, we have finalized the agenda on Friday last week. So it has been really hectic because we put together a large number of invited talks and abstracts and posters, and we received over 80 responses, but we have screened it down, and in the next 2 days you'll be seeing all the presenters and the research discussions. So the objective of this, as I pointed out when we first started, was to bring in the collaborating research partners and think about the future as well, both internationally and locally. So some of the local partners, Sabaragamu University, Eastern University, and we also have some industrial partners as well joining the program. When we started low-key. The idea was not to make this a commercial venture, but rather an event that discusses, shares new knowledge, and inspire the youth who are conducting research. I'm not going to drag on. Once again, thank you very much for turning up. I know the numbers are small at the moment, but as the day goes on, we will have more than 100 participants joining us, and wishing everyone a very successful ICCMMT 2025 and an experience that you won't forget. Thank you very much.
Thank you, sir, for that warm and inspiring moment. We are delighted to have the partnership of Qujing Normal University, People's Republic of China, as the co-organizer of this international conference.
It's now time for Professor Chen Li, Dean of the College of Biology and Food Engineering, Qujing Normal University, to address the gathering.
Occasion of the international conferences on microbes and microbial technologies convened in Sri Lanka and hosted by Rajarata University and Qujing Normal University. I would like to convey our warm felicitations and sincere salutations to all the experts, scholars, and colleagues attending this conference. This international symposium presents a valuable arena for scholars in the fields of microbiology and related technologies to engage in intellectual exchanges. Moreover, it constructs an important conduit for deepening the amity and cooperation between two universities. Despite their minuscule scale, microorganisms possess vast potential to exert a profound influence on human health, environmental sustainability, and social advancement. It is my aspiration that the incisive presentations and the in-depth deliberations during this conference will offer meaningful inspirations to all the participants and infuse new actors into scientific research and talent cultivation in related fields such as biotechnology, food science, and food science in both universities. I wish you a very productive conference with exciting and encouraging discussions and exchange of knowledge so that together we can anticipate a future of groundbreaking knowledge, research, and technology in microbes. Finally, may this conference be a successful and enjoyable one. Thank you.
Thank you, dear sir, for your encouraging words, spirit of collaboration that makes this international conference truly meaningful.
Now, it's time to hear from a distinguished personality who leads with vision and dedication. It gives us great pleasure to invite our chief guest, Dr. Janaka Pushpakumara, Acting Vice Chancellor, Rajarata University of Sri Lanka, to address this distinguished guest.
Good morning. Dean, Faculty of Technology, Professor Bhagya, my colleague deans, registrar, and foreign delegates, academics of the Rajarata University, non-academic staff, students, ladies and gentlemen, very importantly, the delegates who are joining with us online, academics from Qujing University, China. So it's really a very pleasure and honor to address you in this important occasion, this ICCMMT 2025 conference. It's a landmark of Faculty of Technology, not only Faculty of Technology, Rajarata University as well. So these conferences, researchers present their findings, not just findings, their hard work of several months, years, their tears, sweat and everything. They dedicated their time, resources and everything for that, and they come and present here. And this is a forum of brainstorming. People get together and discuss about these findings. So that may be the first step of converting these findings into policies, technologies, practices and all. So these forums are very important. So thank you very much, Dean of the Faculty and organizing committee of this conference, for organizing this event with a lot of difficulties and challenges currently we are facing in Sri Lanka, but still thank you very much for arranging this and make it happen, because to come out from these challenges, these difficulties, one way is finding new technologies, new policies, new practices. So this is very important. So I don't waste your time. So we should proceed with the real science. So this is actually a political talk. So I don't want to take your time. So let me congratulate all the presenters, researchers, and at the same time wish you a fruitful conference ahead. Thank you very much.
Thank you so much, sir, for your encouraging and thoughtful words. And now, we are truly honored to have the Professor J.D. Bhatt from King Saud University, Saudi Arabia with us.
Professor J.D. Bhatt, a mycologist with national and international work experience, has an academic and research career spanning over the last 45 years and contributing to taxonomy and biology of microfungi. Sir, we warmly invite you to deliver the keynote address on exploring extremophilic fungi in less endowed Asian regions and habitats.
Just yeah. I'll have to stand here. Yeah. Good morning, ladies and gentlemen, and esteemed Vice Chancellor of this university. I'm very grateful to all of you to be here. I am formerly from the Department of Botany, Goa University, and presently I am associated with the King Saud University in Riyadh, Saudi Arabia, as a visiting professor there. They call it as a distinguished scientist fellow. I have chosen a topic of my interest. I'm primarily, firstly very delighted to be here at Rajarata University for this international conference. And I bring fond greetings from India. Sri Lanka and India are very close neighbors. Our relations last for more than 2,500 years, just what the history tells us. And this relation between the countries was built on a long legacy of intellectual, cultural, and Professor C.V. Subramanian bought some samples from Kandy and passed on to me to work on the fungal diversity of this region. And incidentally, we collected one new fungus. We named it as Nectria Kera. Kera refers to coconut tree. So that was the first fungus we described from Sri Lanka in 1985. I am also that was described in 1985. I was also associated with the two important mycologists from Sri Lanka, Professor Dr. Shivanesan and Dr. Punithalingam. These two mycological giants, those days working in the Commonwealth Mycological Institute, Kew. When I visited Kew in 1985, these two people were working there, and Professor Punithalingam passed away just 3 years ago, and Shivanesan wrote a voluminous book those days, the bitunicate ascomycetes. I am telling this because Sri Lanka also has a legacy of mycological studies in a great deal, and that is a little bit of history. Whereas presently I am working with quite a lot of Sri Lankan mycologists, and to name a couple of them, Dr. Samantha is here, Dr. Nalin, and Dr. Chaturanga. Couple of years ago, when I passed through Rajarata University to Batikola to attend a conference, I was very impressed with what they are doing here, and we invited Dr. Chaturanga to collaborate on a program, and the part of the fruit of that program is today's conference. So I'm very grateful to Rajarata University from this angle. Okay. Oh, it's going back. I have chosen a topic of my interest for today's talk. I'll finish it within 25 minutes. Fungi in less endowed or extreme regions, extreme habitats, but primarily of the difficult regions in the Asian subcontinent. I have kept this title, but I will be speaking primarily on the fungi, how to infuse the younger people, what we can do with the fungal studies. We know what is a fungus, why we are naming them. I wrote one sentence, why the name for an organ, why I am called Jairam. It's not for me, it is for others to refer me, to recognize or to identify. So name is very important. Taxonomy is revolving around this particular concept. And why naming fungi? Because fungi are so important for us, starting from antibiotics to food. So that is the purpose we need to work on them. Oh, it's going back. Fungi are present everywhere. There are ecological habitats everywhere. I don't really need to refer where fungus is not there. Every breath that we breathe has got fungal spores. So fungi are present in any of the habitats, whether it is in the plains, mountains, streams, everywhere you will see the fungi. And we have chosen for our work some of the habitats which are a little difficult to work with, not really for the fungi to live with. So we'll look at them and the kind of substrates, any organic matter for that matter, we should be able to see the fungi. And how and where do fungi grow? They grow everywhere. Epiphytes, endophytes, plant pathogens, animal pathogens, mycoses. All these are the areas where we'll see the fungi. And Besides this, there are also fungi which are very specific to the substrates. Like if you, the group of fungi which grow on herbivore dung, for example, the coprophilous fungi is a kind of an extreme habitat. All fungi don't grow there. So we will look at them, some of them, and how and why they are growing there. Stored dry fruits, air spora, everything is a habitat for the fungus to grow. Next one. And based on the kind of substrates and the habitats they grow and the kind of their behavior, fungi are called as, they are grouped in different life modes like saprobes, pathogens, endophytes, epiphytes. I have given some pictures of them. There are a variety of fungi. We qualify them based on the life modes. The lignicolous fungi, fungicolous fungi growing on other fungi and so on. Next one. This is Dr. Nalin's paper. I have put this because if you look at the textbooks of some 10 to 15 years ago, we would refer the fungi into just 5 phyla, major phyla, like phycomycetes, zygomycetes, ascomycetes, basidiomycetes, and fungi imperfecti. That's the kind of groups. But as the science advanced and the kind of information that we gathered, today fungi are about 19 phyla. That's the kind of picture presented in this paper, which enormous work has gone to this paper. And I must congratulate Dr. Nalin, who is an amazingly great worker today. That's the kind of, although still those two groups are the huge ones, Ascomycota and Basidiomycota. Next one. This is another exercise which most mycologists, they keep doing. How many fungi are there? Presently we know only about 156,000. That's the number what we know. I mean, that is what we have labeled them by name. Whereas how many fungi are there? Different methods are used, extrapolating this number, estimating this number. And one of the methods they use is how many plants are there. So if we take 1:6 ratio or 1:10 ratio, we will get some broad picture. So they kept this number 2.2 to 3.5 million. That's a number. It's an estimate. In other words, there's a huge gap which we will have to look into using the advanced technology in the course of time. That is what one of the exercises we do. And where are these fungi? Next one. That's another exercise people keep on scratching their head. Where are these fungi? Are they really unknown? No. They are really, we don't know where they are. So again, the kind of exercises which mycologists keep doing is, where are these fungi? So the kind of presumptions we have is probably they are all in the biodiversity hotspots, or some species may not be expressing, cryptic species, or we don't know them where they are. And one of the issues which we are discussing in our collaborations is there are many little or less studied habitats. So if we look at them, we may get some fungi, we might get, and this is what we are doing, and probably they are really there, some of them. Little or less studied regions. That's an issue which we will look into. Next one. Yeah, but so if you look at the Indian, the subcontinent here and Sri Lanka, both these areas are the biodiversity hotspots. So one of the issues, probably this is where the diversity, high diversity is present. Next one. press a couple of times. Yeah. I have taken a couple of 3 or 4 habitats, probably less endowed or less studied. That's what we look into. One of the areas is the aquatic fungi. Fungi living in water of any kind. They are present in marine, freshwater, again within the freshwater they are present in running water, standing water. So that is one of the difficult environments to live with. But are there fungi? So we did some work in the early days in 1970s, 80s. So it's not well expressing. We did in 1979, 81, those years, I had an opportunity to scan the fungi in the Western Ghats region of southern India. That's the project which my professor had. So one of the assignments he gave me those days was look at the aquatic fungi when you run through these areas. So what I did was I took few vials, small vials, and one small bottle of FAA, formalin, acetic acid, alcohol mixture. So wherever I went, if any streams I passed through, I would collect the foam sample, put few drops of FAA, and put those bottles in my bag and bring them back to the lab, examine those foam samples. I did this from the central region of Western Ghats, from the top, Mahabaleshwar, down to Kanyakumari region, Cape Comorin, this region, until Rameshwaram, the entire stretch, or 1.5 years we examined this region. And fascinating fungi we saw, and we wrote one paper, Conidia from freshwater foams. That is a paper which is very poorly expressing. In 1981, we published this paper. And that happened to be the first study of aquatic fungi. But those days, we really knew very little about the aquatic fungi. Now, when we fixed these fungal spores, why we fixed them? Interestingly, these fungal spores were very characteristic with this, either they are appendaged, or they are branched, or they are variedly coiled. That was the structure we had. And those days, late Professor Webster, he wrote me one letter. Not those days, we had no email facilities, of course. And he wrote me that, but you must fix them because spores are very characteristic, and if you don't fix these spores, within few hours time, these spores will settle down and they will start germinating. So you will not be able to really identify the fungus. Perhaps those days, this was one group of fungi where we could identify the fungal species merely based on spores. No other group of the fungal kingdom where you can identify the species looking at a single spore. So, Ingoldiella hamata, for example, here, that fungus was very characteristic with this branched conidium. This one. So, that kind of exercises we did, and it was only a very casual observation. But what this paper did was gave an enormous enthusiasm to work on this group of fungi. So, next one. So this is the kind of exercises I did. Those pictures I took much later, only for the preparation of a slide. But I was quite younger those days, went around and collected those samples, foam samples. And if you examine the foam after coming back to the lab, you would see this kind of slides where spores were we could examine them. So the kind of foam, and if we had identified that areas, we would get a picture of the fungal diversity of that region. So we did that those days. Next one. This is the current exercise what we do. Not only we collect the samples, not only the foam sample, but also the substrate, plant substrate from the given region, bring them to the lab, look at the fungus after incubation for a few days, then we also isolated those things in culture. So, because culture is a starting point for any biotechnological exercises later on. So this is the kind of exercises we do. Next one. And once you have the culture, you can undertake the molecular sequence work. This exercises we do presently in collaboration with this university. I will come to the picture what we have, the kind of results we have. Next one. Not only the saprobic fungi, but also we worked on the endophytic freshwater fungi. Those fungi found in aquatic plants, internally growing them. Look at the kind of, this is why we call them as extremophiles. In an extreme environment they live, these fungi. Next one. The present picture of fungal diversity is something like this. In 1975, Ingold wrote a book merely looking at the fungal spores. And those days, the total number of fungal species were only 98. 98 species were known those days. Looking at the diversity across the entire globe. Whereas presently, that's the number we have, around 3,000 species. And the amazingly large diversity, and where the highest number is in the Ascomycota group, 2,900 is in 2022. That's the kind of picture available. And within the Ascomycota, next one, Solidariomycetes again it's a large number, 823 species. Then Dotridiomycetes, second group, and these are the different numbers we have within the Ascomycota. So that is the group which is occupying a central position in the aquatic fungal diversity. Next one. I put this picture just to tell you the fascinating, to look at under the microscope, these aquatic fungi, especially the Ingoldian fungi. Each fungus, you can identify them merely looking at a couple of spores. Next one. We surveyed the freshwater fungi over the years to not only in the Western Ghats region, but also northeastern forests, Andaman Islands. Main interest was to see what kind of aquatic fungi are there, number one. Mostly the Ascomycota. Only the Ingoldiella hamata, that is the species from the Basidiomycetes known those days. And these were some of the early papers we published in the, everything was merely looking at the freshwater spores. Next one. But in our recent exercises, after initiating the collaboration with the Rajarata University, with the Professor Chaturanga, we have some very interesting observations on aquatic fungi. I'm more, grateful to both Nalin and Professor Chaturanga, because if they had not taken this, the understanding and interest, we wouldn't have been successful in bringing out something, very fascinating area of science from this region. What it tells us, leave these papers alone. It's like a gold mine. You collect the samples, you will see some fungi. That is one. If we get something new, good enough. Even if we don't get, I have a paper now, another one, which we published in the Frontiers just few days ago. It has come from published. What it tells us is, once you have the culture in hand, and that fungal culture can be worked further, what kind of enzymes, what kind of metabolites they have, and what we can do with them. That's the kind of work, and why it is happening is because this is an institute where you have the faculty of technology with various other departments. This morning I was talking to Professor Chaturanga. He said, once we have the, once we knew that this fungus can decolorize, I have in a couple of slides later. Interestingly, that species, the enzymes produced, could decolorize the synthetic dyes in effluents. So that's a kind of pollution control can be worked out using the fungal principles. So not only we did these basic exercises, but he went ahead and developed a protocol, what we can do further. That is the beauty of this kind of work. Next one. You can see the kind of diversity looking under the microscope. The freshwater ascomycetes, variety of sexual morphs. Next one. Asexual morphs and sexual morphs. It's a diversity not only in this, even within a fungus you will see, within the genus, so much of diversity. All we require is young students to look at them with enthusiasm and interest. This is a message I want to give to young people, that science is the one where you should work with enormous interest and curiosity. Curiosity should be the driving force. Next one. Look at the kind of diversity in their structure. Next one. I put these slides to show the amazing diversity in their fruiting structures. Next one. And certain groups of these aquatic fungi also are now explored for their bioactive principles. Next one. Yeah. Go ahead further. Yeah, this is the one which we in front, this one and the next paper. These two are the ones which we recently published from, so I am thankful to these people, especially my colleagues, Dr. Professor Chaturanga and whole team of them for this amazing work. In vitro evaluation of synthetic dye decolorization by filamentous ascomycete isolated from freshwater environments from here, and development of a prototype for addressing the environmental pollution from synthetic dye contamination. A long title we wrote, but that is the joy of this work. You should, all of you should take time and read this paper for the kind of information we have put together. I am only a passenger in the bus, but the drivers and the navigators, all these are the people here. Next one. That's one area of extreme environment. Second one is the mangrove habitats in the coastal region. It's a difficult environment where any organism to live in, and there are interesting fungi which my colleagues from Goa, they are working on. They have many fungi which are they isolated and published during the course of time. Next one. This is another area which also very interesting, which we should explore even in Sri Lanka. This is one done by my colleague, Dr. Ashish and Pratibha, both of them. They were my former students, but my colleagues now. And this fungus, Acrodontium, this species, if you look under the microscope, it is just an acremonium, very simple one. But when we investigated the molecular sequence data, we found out this to be an interesting fungus growing on insectivorous plants. Very difficult environment, but the fungi grow in that pitcher traps, liquids. Next one. This is another fungus which we collected in kokum, one of the spices tree. Interestingly, this fungus was growing only in those regions where insects have already corroded the plant, burrowed the plant. So the fungus grown only on insect infested regions on the leaves. That's an interesting findings we found out. This Caliciopsis, that's a region where the insect first ate the leaf, and that region only the fungus was found growing on them. So this is not an easy, we really don't know why it's happening, but it's happening. Next one. There are fungi associated with the medicinal plants. There are people working in Thailand, in China, especially looking at the endophytes growing on medicinal plants. Next one. There are teams of people working, exploring to find out, are these fungi interesting ones? Why they are growing or associated only with the specific plants? That kind of information. Next one. Besides this, we also did an exercise looking at the group of like palm trees. Are there some interesting fungi? All you need to do is to explore them. You will certainly, get stumbled with some of the fungi which you have never seen earlier. Next one. Similar to palm, then this is another plant which Dr. Samantha and the team have worked on this. On mango trees, taxonomic and phylogenetic studies. I've just put this, where all you can look for fungi. The extreme, is it really difficult areas? Instead of looking at them in general, look at them with some focus. Then you will come with fungi. Likely they will have some very interesting metabolites. Next one. There are areas still, some of the less or difficult areas like sand dunes, coastal sand dunes. Next one. Coprophilous fungi. I said little early, in the beginning, this is another area which should be a focus in the coming days. We did some work in India. I'm sure any country this can be taken up, because or any region we can take. There will be definitely, you will be found coming with different groups of fungi, and it's very fascinating, this particular. I'll take only 1 minute here extra. If you look at the fungi growing on herbivore dung, why fungi really come up on dung? These fungi are very intimately associated with their life cycle with dung habitat. Plants, these herbivorous animals, they feed on or graze on, especially on the grasses and herbaceous plants. And those spores which are shot out and sticking to the leaves, they are passing through the gut of these animals. And one example is Pilobolus, this fungus. And this fungus has got a sporangium, And it shoots its sporangium. A microscopic organism can shoot its sporangium about 3 meters height vertically. And this kind of adaptation this fungus has developed over the years through evolution, primarily for its survival. So the spore which is sticking to the leaves are ingested by the animal, herbivorous animal, and only when it is passing through the alimentary canal or the digestive juices treatment, the spore germinates in the next generation. So that's a kind of not only the mucораceous fungi, even Ascomycetes soldaria, that's an example, or Coprinus in the Basidomyces. All these pass through the alimentary canal and then only they digest. So they come in succession. In days, within 15 days time, the entire dung gets decomposed by these fungi. So very interesting area. And we did some exercises with she is here, Dr. Nam Som. She did some work on the dung decomposing fungi from Thailand. And very interesting fungi, they have some lignocellulose decomposing fungi we isolated from them. Life cycle, and this whole group is very interesting. Coprophilous fungi. Next one. I only fused it. And the cave, for example, work done by Dr. Fai. Cave is an interesting and very difficult place to live with. But there are fungi growing in that difficult environment. If a fungus living in a difficult environment, isolate them and examine, it's a kind of an intuition I have that they will definitely have some very interesting metabolites for our future. Next one. I have only a couple of slides more. There are fungi growing in specific substrates. We don't know why they are growing. Both its asexual morph and sexual morph are growing side by side, but we are not able to culture them. But if we are able to culture them, probably we'll know why they are growing together. So that's a great big area of interest for the coming, in the next generation to work with. Their spores, they don't germinate in culture. So we really need to know why they are not able to, we are able to culture them. It's an interesting group, very intriguing too. Next one. Fungi also grow in the air. My colleagues from Pune, they went to Mahabaleshwar, exposed some petri plates in the open air, and they found out some fungi, and when we examined them, found to be very interesting, and we named it after Stan Hughes. I have put this slide to tell that he was one of the giants, lived for 100 years. He expired only 2 years ago, and in honor of him we call this fungus as Fusicola hughesii. Very interesting fungus. Spores were isolated from air, and we haven't found them anywhere otherwise. Probably they are growing in the forest habitats. Member of the Nectriaceae group, Ascomycetes fungus, very interesting. So fungi are everywhere. We'll only have to look for them. Next one. Yeah, there are several such examples, habitats and habits. We'll only have to examine them. One area which should be our future focus should be the gut mycota of both invertebrates and primarily the invertebrates. I often see sometimes very tiny insects crawling around in some decomposing or decaying wood and trying to burrow them further. How it is possible? Some of the most difficult environments they are able to. So that is because of the gut mycota. So anaerobic partially, but it's worth exploring them. If we can get them in culture, they will be the for future for us. Herbivore and endophytes of plants, the various plants. There is one paper here in the course of time. You'll see fungi from the ancient historical buildings. Probably they are living there for a long time. If we put them in culture and see what they are doing and how they are surviving is worth an area of interest. Then I often feel that plants growing in steep escarpments in the hilly terrains, we should explore the endophytes of those plants because they will have something very interesting which we don't know so far. Every habitat, every field, habit should be our focus in future. Next one. I think this must be the last slide, yeah. We are presently collaborating with, I said, Professor Chaduranga and Naren with this university. We are at the moment focusing on freshwater streams, ponds, thermal springs, marine waters. That's what we are doing. And we do this work with enormous care and respect. Care and respect. I mean, I also a firm believer that any of these materials should not go out of the country because that is the law and we should respect them. So nothing like working in the same place and developing, and this should be useful for the region in future. Similarly, in India, we work in the mangroves and salt marshes in Goa, where I'm living. So the ecological systems are similar, and we will have a great. It's not only for us, for our future. So we should explore this fungal diversity with more enthusiasm, and not only explore the fungal diversity, but also look at their chemical creativity, what they can do for us for our future. So that's what I wanted to tell in this presentation. And very thankful to all of you for your patient hearing. And I want to specially place on records my thanks to King South University, where I am presently working as a distinguished scientist fellow. And because of that, this program, I could participate. This collaboration is primarily with the support from that program, and my colleague, Dr. Turki is here. And we did a great deal of discussion how we will work it out. I said this grant should be available for somewhere. So we did this enormous work went on to this. So I thank all of you. Not to explain further this, I'm very grateful to this university for providing the space for this work, and personally thankful to everyone here. And such an exercise gives, I am not young, but it gives a lot of enthusiasm and energy to work on such places. So thanks for every one of you here. And where is Sam? Yeah. Thankful to you, Sam, because these are the my close friends with whom I have been working last 15 years. So thanks everyone. Dr. Nalin, thank you very much. So Mr. Vice Chancellor, it's very nice of you to sit here patiently. I know all of you are very busy, but you took your time to be here. Thank you very much. Thanks to every one of you. Thank you. Yeah. Sure. Thank you very much. You can close that.
Thank you, Professor Bath, for that fascinating and inspiring talk. What an amazing insight into microbial exploration.
Next, let's take a brief journey through the inspiring story of Rajarata University of Sri Lanka, proud of academic excellence and innovation.
Rajarata University of Sri Lanka, located in the historic city of Anuradhapura, was established on January 31st in 1996 as the 11th National University in Sri Lanka. For nearly 30 years, Rajarata University has shaped minds, careers, and futures. Today, it is home to more than 12,000 students and 6 faculties, namely the faculties of Agriculture, Applied Sciences, Management Studies, Social Sciences and Humanities, Technology, and the Faculty of Medicine and Allied Sciences, each possessing a distinct academic identity and mission. Rajarata University has strengthened its global presence through partnerships and collaborations with international and local institutions, while also securing competitive grants to advance cutting-edge research. Rajarata University is more than a place to study. It's a space for learning, discovery, and collaboration. Over the past 2 decades, Rajarata University has transformed into a leading force in higher education and scientific discovery through research. During the last 5 years, RUSL researchers published over 600 Scopus Index papers, and nationally, Rajarata University shines with researchers receiving prestigious presidential awards for their groundbreaking work. With these outstanding contributions, the Research Excellence Award ceremony of Rajarata University was also initiated in 2022 to motivate a culture of celebrating academic achievements. The university hosts international and faculty-level research conferences every year, creating platforms for students, academics, and global scholars. The university's achievements are recognized worldwide. Simigo ranked RUSL first in Sri Lanka in 2022 and 2024, proving its consistent leadership in research excellence. Adding to this pride, several Rajarata University professors and alumni have been ranked among the world's top 2% scientists by Stanford University and Elsevier, an extraordinary recognition of individual and institutional strength. Celebrates this spirit, recognizing outstanding research across diverse disciplines, deriving from both local and global experts. To Together, we move forward. Research, publish, inspire.
We have now arrived at one of the most exciting moments on this inauguration ceremony, the launching of the conference proceedings book of ICCMMT 2025.
This publication is a proud reflection of the collective academic efforts and scholarly contributions presented at the international gathering. It encapsulates the essence of knowledge, innovation, and collaboration shared by the researchers from around the world.
May I now kindly invite Professor J.D. Bath, King Saud University, Saudi Arabia, Professor Samantha C. Karunaratne, Qujing Normal University, PR, China, Professor Chaturanga Bamanwarachige, Rajarata University of Sri Lanka, editor of the proceeding book, Professor Nalin and Vijayawardena, Qujing Normal University, PR, China, to officially launch the conference proceedings book. Thank you very much, dear professors. This book symbolizes the dedication and research excellence of everyone gathered here today. Congratulations to all the contributors on this milestone.
Every great event deserves a moment of gratitude. To deliver the vote of thanks, I cordially invite Professor Nalin N. Vijayawardena, coordinator of the ICCMMT 2025.
Good morning, everyone. It's my great privilege to deliver the vote of thanks as a conclusion remark of the inauguration session. So on behalf of the organizing committee, I wish to express my heartfelt gratitude to our chief guest, Dr. Janaka Pushpakumar, the Vice Chancellor of Sri Lanka. And I would like to thank all of the faculty members, non-academic staff from the organizing universities, which mean Rajarata University of Sri Lanka and of course Qujing Normal University, PR, China. And I especially thanks to our all foreign delegates who attended this conference, and all the presenters, of course, and it's very important all of you to be here today. And then I should thank to all the faculty members and our panel members from other universities, and of course from the other faculties of the Rajarata University. And we should extend our thank to all the reviewers and editors of our abstract book. And we should thank to all our partner institutes that their sponsorships, their contributions are really, really helpful for us to make this event true and to become everything success here. And then I should thank to all keynote speakers, invited speakers, plenary speakers, and panel members who are coming here physically and who will attend during the online sessions. And of course, I should thank all the contributors who contribute with their research findings today with our research conference. And of course, I should extend our thank you particularly one person, actually it's two persons, Professor Chaturanga sir, the Dean of the Faculty of Technology, and one particular person, Mr. Bhagya. He is the person who behind most of this work. And there are some other person that I cannot name personally or here one by one, but all your efforts, it's very warmly thankful, appreciated by our organizing committee. Thank you so much.
Thank you, sir, for those heartfelt words. And with that, we have reached the end of our ceremony. But this is just the beginning of 2 exciting days ahead.
Absolutely, Lumini. Let's continue the conversations over a cup of tea. You are all invited to join us for tea and networking at Bite Cafe at ground floor of this Epiphany building. Before we head for the tea break, we kindly invite all our guests, speakers and participants to come forward for a quick group photo.
And after the tea break, don't miss the roundtable discussion starting at 11 a.m. in the auditorium.
And finally, thank you for joining us this morning. Stay curious, stay inspired, and have an amazing conference ahead. And ladies and gentlemen, let's stand for the national anthem.
real-world applications, emphasizing the synergy between scientific discovery, innovation, and industrial implementation. We are joined today by an outstanding panel of experts, each contributing a unique perspective to our theme. On my left, we have Professor Samantha Chandranath Karunaratne, leading researcher in mycology and specialist in mushrooms. And then we have Professor Jayarama D. Bath, distinguished professor in microbiology and plant pathology. Then we have Professor Chaturanga Bamnaarachige, researcher in biotechnology and microbiology, who is also dedicated in industry-academia collaborations. And last but not least, we have Professor Nalin Vijayawardena, whose work focuses on advancing research collaborations. Today's discussion explores how we move from the basics of microbiology to its many applications in biotechnology, agriculture, environmental management, and beyond. To start off, this question is to all of you. What does from basic microbiology to applications mean in your respective field?
Thank you very much, Dr. Chaturi, for the introduction. So I am honored to be in this roundtable discussion. So this discussion is very important because in my field, the mushroom taxonomy, phylogeny, and domestication. So we should not only work on the taxonomy. The taxonomy is important. Like if you don't do taxonomy, we don't know what we have in any country. And don't stop in this taxonomy only. So go beyond that. Do something for the community, and especially for the farmers. So what we do in our research, we can pass to the farmers. And then we have some value-added products. So this is what we need to do, like not only the pure research, but go beyond that to the applied research.
Thank you, friends. Yes, it's a very interesting and very important session that we have. Discussions are primarily to understand the subject beyond its limited description of any area, whether the microbiology, because when we studied or when we were young, taxonomy was said to be very important, and it used to be worked in isolation. When the basic sciences extended beyond that to applied sciences, I used to feel that it is only science and application of science. So we have examples from even from earlier days, like Alexander Fleming, when he discovered the antibiotics, said to be a very chance discovery. But that one discovery those days, and it was also done out of compulsion, because during the First World War, really they were looking for something beyond the simple antimicrobial agent for the kind of problems they were having in the field. So antibiotic was, though it was a chance discovery, but it led to the discovery of numerous other programs, principles later on. So that was the starting point. So taxonomy also is just a starting point. In the course of our discussion, I will come to some specifics later on, what we can do and how our science should focus later on. So very thankful to you for the inviting us, and we will really make it an exciting discussion in the course of time. Thank you.
Thank you, Professor Bhatt, and thank you, Chaturi, for starting up with such an interesting question, which is multifaceted to say the least. And among the distinguished scientists, I'm the odd one out. I'm not a taxonomist, I'm a molecular biotechnologist. But as you said, it's very important to have the basics, because once you cover the basics and when you start going into applications, there will always be issues. The only way you can handle the issue is by the depth you have covered at the basic level. For instance, if we are discussing mode of action, mechanisms, gene expression, and how you control, and even when you look at taxonomy, I mean, there are various sectors. For instance, are we thinking of multi-loci phylogenetics? Or that way we might identify subspecies levels that may differ from each other, again, which would allow us a pool of genes to work with. So the basic science need to be covered in depth before we move into applications. Of course, we can move into applications with a little bit of basic sciences, but we will definitely run into quite a lot of issues unless, of course, we know the depth of our basic sciences. That's my feeling.
Thank you, Dr. Chaturi, for the introduction. And it's a great honor to be with this excellent scientist. And of course, I am, as Dr. Chaturi introduced, I have mainly my experience on the taxonomy of fungi. But of course, now, as Professor Samantha mentioned, that we are trying to focus on not only taxonomy, we are trying to focus on how we can use these fungi in real-time applications. So that's very important. And of course, we will discuss about this little bit detail. As all are saying that we need to do our taxonomy part very carefully at the very beginning. Otherwise, we all will be in trouble. Simply, it's a good example in these days we are talking about antibiotic resistance, and then maybe you have heard about some fungi, they are resistant for some antifungal medicines. So in these cases, we have to identify the causal agents very precisely at the very beginning. So that is the job of the taxonomists at the very beginning of any field of science. So still somebody says that the taxonomists will be an extinct species in the future, but I don't believe it. So, of course, we have a lot of things to do as taxonomists, but still it's a very important and the basics for each and every sciences.
Thank you so much, everyone, for setting the scene so well. My next question is for Professor Bach. How does transitional mycology still drive innovation today?
Traditional mycology.
No, transitional.
Yeah, transitional.
Yeah.
Okay. See, science, when it, I also, they sent to this agenda prior to the meeting to us. So I was looking into these questions, what you have put there. It's a good area of understanding the subject. Science is nothing the recent one. Science, indeed, traditional microbiology. Is there a traditional microbiology? I wanted to say. For example, alcohol making or curd making. It's nothing new. It's all new from the time memorial that these things are part of our day-to-day living. So we are only refining them further. So the traditional to transitional to the modern, it has to go like that. So we must look into this and put our science head together and go really what is required for today. We have very serious issues, environmental issues, which really requires inputs from the basic science. There are issues with the climatic changes. There are issues with the pollution. Requires some food crisis. Everywhere we require science. And there is the role of microbiologists and mycologists specifically are a great deal of newer inventions are in the course of time required. I think that should be the transition we require in the future. So we look forward for such developments in the time to come. Yeah.
That's a great explanation. Thank you so much. So every breakthrough in applied microbiology begins with a solid foundation of basic sciences. Professor Bath, again, what are some unexplored areas in basic fungal biology that could lead to new applications, in your opinion?
I think unexplored areas. I have made it this morning some references of where we should look for. Okay, as a mycologist, we go to some specific areas I explained this morning also, where we will look for. Now, I won't put this as a newer fungus, but creative fungi. Creative meaning what kind of application we will have from that fungi. This is simply an imagination I have in my mind, which I have put into exercises course of time, like the difficult environments, aquatic fungi, one of the areas, and we'll take only one issue. Why some fungi grow in specific areas? What makes them to grow in such an environment? For example, there are fungi growing on human hair. It's not easy for a fungus to grow in such an environment, such a habitat. So if we explore or go further science, we kind of explore them further, we will be able to get some fungi which will have some creativity with us. That is one aspect. Second, if we look at some of the fungi growing on medicinal plants, we have good examples of like Taxol. At one point of time, 1990s, people looked for the Taxol, all kinds of trees, endophytes, they searched for some fungi, especially for a Taxol producing, because that was found to be one of the cure for cancer areas. So it's not only one group. I would say we should look into all kinds of group-wise plants, medicinal plants, or even phytopathogenic fungi. We don't know them. For example, the Philostricta. There are many species of them now producing Taxol. So we must look into every fungus for that matter. We should explore we have the recent, our own studies showed very ordinary fungus, it was Fasciodiplodia species. But we found to be one really creative one. So it is my intuition and firm belief that every fungus is a standalone. We must explore them further, and we should invite people around, as Professor Chaduranga did with this fungus, from the biotechnology area, from what are the other groups you involved? Material science. So we developed a very fascinating prototype on this. I think that is a You really should highlight this at the government level, should be able to get them further in the course of time. So science and excitation, it's only with us. If you don't show this excitement with this kind of exercise, we will not be able to do further. Yeah, thank you.
Thank you so much, Professor Ba. Professor Samantha, perhaps you can answer my next question. How do studies on fungal physiology or ecology support applied uses like cultivation or medicinal production?
Yeah, so in order to understand how a fungus or mushroom works, or how the fungus behaves in the environment, we need to understand the physiology and ecology of the fungus or the mushroom. So if we understand this well, so then we can apply this to our applied research. So for example, if we understand what kind of secondary metabolites a fungus secretes, then we can use these specific fungi to extract certain enzymes and like antibiotics, for example, this penicillin from penicillium. So this kind of secondary metabolites can be used in the industry. So in order to use these special fungi, we need to understand their physiology. So physiology is very important. And also, if we want to cultivate some mushrooms, we need to understand their ecology, like what kind of temperature these mushrooms start to fruiting, and what kind of substrate they prefer. And if we understand these things well, then we can apply these to our cultivation processes. So these are some examples, but I can give many examples, but since we have limited time, so I think you can understand how this physiology and the ecology are important for the applied research.
Thank you so much, Professor Samantha. Moving on to Professor Nalin. What steps can institutions take to strengthen the capacity for fundamental research?
I think it's very important, and we cannot address that type of thing very superficially. First of all, we, as per Samantha and Professor Ramsar told, we need to understand the depth of the research and what is the outcome of your fundamental research. So based on that aspects, we have to link different institutes, of course, with the different human resources. I think at the moment that is one of the important limitations in Sri Lanka at the moment. We know that most of the masterminds, they have gone out from the country. So then we are in a big trouble how to get the best minded brains to link with the research. So that's and of course then the institutional level, the people should welcome the other people from the outside the country. That's also a very important thing. And the people should be very humble to welcome other people. So good example is particular technology faculty. You guys welcome all of us. I mean, and there are several delegates over here. So there are no any limitations. That's very important thing, as far as I believe, because we have a lot of experiences when we trying to start work in Sri Lanka. So, and the other thing, on the other hand, when we decide the plan, the architecturing part of the research, I strongly believe that we should link with some government people. I mean, policymakers. Of course, the policymakers can be the top level to the bottom level. The hierarchies will be very important. And of course, we invited last few days. We invited some people, and some people joined us today. So it's very important to link this level when you planning and architecturing your fundamental research to any kind of applied research. Yeah.
Perfect. Thank you so much, Professor Nani. So moving discoveries from the lab level to real-world applications is where many challenges arise. This is also what we call the transitional gap. Professor Chaturanga, what are the biggest barriers when translating lab research into industry-ready solutions?
Yeah, to begin with, Chaturi, we have tried several models. So basically trying to move from our basic research into applied solutions. But what I have realized is all of us seem to work in silos. So there need to be a connectivity among different stakeholders, including the industry and the surrounding communities, maybe local communities, and also people who face the real life challenges. Because sometimes we get carried away when we work in the lab, when we see a new strain, when we see a gene that interests us, and expression levels that we check with using our technologies. Maybe we go for RNA-seq, we go for QRT-PCR, and we are so happy that we have identified something that works at the lab level. But when you take it to the world outside, it has to address some of the key issues in your community. Unless otherwise it could be your fancied research that may have very little output. So we first tried with this model of using our cultures and looking at common enzymes like alpha amylases and cellulases like that. But of late, we are connected with the industry, and we see that the industry take us to a totally different world. When you talk about trying to move the lab research into application, one of the biggest issues is the focus. Because in terms of getting funds for developing applications, if you look at any funding source, the number one interest is whether it is handling a real life situation, whether it has enough potential to make an impact. Because I'm also serving as a reviewer for biotechnology proposals for the last 20 years or so in the two leading granting societies, the NSF and the NRC. So they are what we really look at when a transitional research comes is whether they have covered enough depth to move into the application side, and if so, whether they have a solid plan to make something impactful. So from the grant side itself, there can be limitations because, for instance, in Sri Lanka currently, no matter how good your research looks, you are not funded for genetic engineering. It is the country's policy. So that could be totally different in another country. Like, for instance, India could be far more receptive to genetic engineering and gene editing and things like that. But in Sri Lanka, one of the biggest issues is getting funding when you're developing the technologies. So you need to understand what makes a bigger impact. So when we started working with the industry, we have seen that they have very critical issues that need to be handled. Perhaps such critical issues could make a big difference. For instance, nowadays we are looking at the vanilla curing, which is a very conventional process. And we are trying to mix in material science and also as he said, the secondary metabolites, because they play a very big role. So the problem here is whether you are having enough vanillin content, as an example. So the current practice tend to make it so inadequate. So we are looking at controlled environments to have better expressions, and also looking at the enzymes. So this is something we can do without moving into high technology. Of course, we could be using technologies like UPLC, MSMS, these kind of techniques. But in terms of handling the genome, we are being very simple about it. So in a country like Sri Lanka, you need to pitch at these critical levels. Otherwise, your research will not move on to the application that we desire. It has to make an impact.
Thank you, Professor Chaturanga. And following up on what Professor said, my next question is to Professor Nalin. What steps can institutions take to strengthen the, what support mechanisms can help researchers bridge this transition gap?
It's how to answer that question. I'm a little bit like this. I mean, for me, I believe, as I told earlier, as an institute, they have to be very humble. I mean, to accept some other people's opinion. Right? I always say that the science can be criticized, but cannot be ignored. Right? So maybe the way we, I mean, in Sri Lanka, some people have, they might have some outdated ideas. Not only Sri Lanka, I mean other countries as well. So maybe you have to be very humble, and you have to be very, of course, humble. To be humble, there is accept or follow others' opinions. So for my understanding is one of the most pioneer point for a research, especially when you bridging some relationships with other institutes, of course, with the other resource person. Otherwise, it's impossible to continue or maybe initiate any kind of fundamental or even a new applied research. But on the other hand, you should have good enough fundings and infrastructure in your institute. So we know it very well. At the beginning, we didn't have good enough facilities in this place for mycology research. But I know how Professor Chaturanga sir and Dr. Soisa is not here, and Dr. Ajit, how those people worked hard to build this place with molecular lab, pathology lab, and they have algal lab, right, and virology. So I know how they did work hard to build this place. So of course you should, for any kind of science, of course you should have human resource, and then you have funding, and infrastructure is also very important. Otherwise, if there are many people like Professor Samantha and myself, even Professor Bhatt, if you like to join to your work team, but if you don't have enough facilities, it will be a huge drawback. Yeah.
That's a great point, Professor Nalin, because this naturally leads to the question of collaboration, how academia and industry can better work together. My question is again directed to you. What models of collaboration work best in your opinion? Models of collaboration?
I believe, of course, we should have a better understanding each other. Still, I believe we should collaborate and try to expand our relationships within our comfort zone. I still believe that, because you can have a lot of friends, a lot of collaborators, a lot of colleagues. So if you see our next sessions, we have a lot of collaborations all over the world, in different places, everywhere. But when you talk about the research, I mean applied research, then you have to be very maybe picky, right? So because sometimes, as I told earlier, we can plan. Okay, we can do like this to the industry. We can do like this for the community. This can be our outcomes, output. But maybe sometimes if you cannot facilitate the proper infrastructure or proper funding, everything will be gone. So at the moment we are planning. We're both planning one more research in the future. So in next 2-3 months probably. But he's keep telling me, Nalin. So we need to think very seriously whether we can meet the outcomes and outputs as the funders expect. So it's still, I believe it's better we keep everything in our comfort zone. Yeah.
Thank you, Professor.
Can I add to that? Because these days we're having discussions with the industry And we have the Innovation Center, Pulse, which is now about to take off. So I'm looking at how the industry look at this venture. And already we have several industrial partners on board. So when you talk about models, there are different models. To be honest, when we first started, we thought of having this portal which can capture issues or problems from the outsiders, just like a chatbot, so that within the faculty, teams can identify these issues and they can propose their own solution. So that was our starting model we had when we first developed the proposal for the faculty. And then we moved on to another level because we felt that sometimes in Sri Lanka, models like that do not work. And when you invite people to come to your place also, that doesn't work. So the best way, like Prophet Muhammad did, is to go to the mountain. So we went to Colombo and we had several stakeholder meetings where we captured most of the issues in our curricula and also in our research. That gave us the platform to have one-on-one discussions with the industry. And they have also had some perspectives of their own. I mean, in Sri Lanka, they have had enough interactions with the universities. Sometimes it tends to make them little uncomfortable. As he pointed out, it all depends on your comfort zone. So one of the biggest issues they raised was the academia is not welcoming when they discuss their issues. And so we opened up, as he said, we opened up and we have started having discussions with the industry. Sometimes you need to be a very good listener to pick up on issues that you can work collaboratively. And at that time, we felt it's good to have a wider range of capabilities. Something I might miss might be picked up by Damit and his crew, or maybe by someone who's working in ICT, like that. So you need to have these people coming from different fields to be involved with these discussions. And now we are moving to the next level, which is basically picking up these issues from the industry and then having an open session where groups can look at it and they can produce the concept note and then move into simple research and then if it is successful, prototypes, and then move onwards. So there are different models which may work for different institutes. But for us, what we felt was this way of capturing is very important. The model we tried first failed. That is just through a website, we try to connect, but this works well for other universities around the globe where they capture these issues. So now we have moved into this model and we also already have an advisory board. 5 industrial participants are in our advisory board. And our next step would be really interesting because now we are working with the university business linkage as well, which is the one that decides on product development, services, and things like that. And they have different models with which we can use. Some could be just assignments. An industrial person comes to you and give you an assignment to complete, or some could be a fully developed MOUs where 2 parties are involved in IP rights and everything. So there are different models, but to begin with, as he pointed out, you need to be open, you need to be a very good listener to pick up issues, and then you have to have a multi-talented team to bring out the solution.
Thank you, Professor Chaturanga. And as a molecule biotechnologist, my next question, how do you identify industrial challenges that can be solved through biotechnology?
Once again, I can take a few examples that we are handling at the moment. One issue is flavor profiling of
some of the artisan tea in Sri Lanka, handmade tea. There's a big market for it because they tend to come out with products which are very unique, but we need to give them the science behind that. It's one thing to say something tastes superior, it's another thing to explain the science behind it. So if you know the science, you can repeat it and you can value add, you can do various things. So this particular group, we are now currently, as Professor Samantha pointed out, we are looking at their products' secondary metabolite profile, trying to see where they are different from other tea and what makes them unique. So there are different ways we can take up their issues, and maybe sometimes all what we need to do is just point them out how the science behind it works. So then we can also look at the external factors, like when he was discussing about mushrooms, he said that it's good to know the ecology like that and why their product is very different, the temperature changes and all these things we can look at. But as a molecular biologist, I think it's a very hard life in Sri Lanka. So I actually changed track somewhere down the line because I was a hard genetic engineering person. I changed all my research into more meaningful in Sri Lankan context, more meaningful research because it's very difficult. Of course, you dream about increasing gene expression and controlling it and using some of the new fascinating tools of genetic editing and all that. But currently, there is a limitation when you use this. And even in the world, there are different perspectives about these new techniques. So you have to understand what is possible within your scope and move towards such research. But at the same time, there's a lot we can do in biotechnology because now we are looking at the genomes, and then we are also looking at microbiomes, and we are looking at how we can improve soil health, how we can improve animal health, looking at the gut microbiota. So we can bring in the fascination when you study these and of course make sense out of it. In the conference itself, we have included several such talks where I remember there's an invited speech on animal gut microbiota and improving the conversion rates, like when you talk about cows and what we can do with the probiotics, because the world is moving away from toxic inputs. So because if I don't know whether you have seen this, but there's a very big move towards cows which are healthy when they produce milk. So we can do a lot using microbiology, which is simple biotechnology, without using genetic engineering. So it's frustrating to be a molecular biologist, but there are few rays of hope where we can contribute.
Thank you, Professor. So we are talking about industry and academic collaborations. Professor Samantha, how can small-scale sectors such as mushroom growers benefit from research linkages?
Thank you,
Thank you,
Chandri, for the
Chandri,
question. So this is
So this is
very
very
important,
important
even in Sri
even in Sri
Lankan
Lankan
context,
context,
because
because
when we
when we
do
do
research
research
on
on
mushroom
mushroom
cultivation,
cultivation,
we first
we first
do
do
small
small
scale
scale
research
research
in our
in our
laboratories.
laboratories.
So
So
we should
we
not
should not
stop
stop
our
our
research
research
in this
in
level,
this level,
because
because
sometimes we see
sometimes we
like
see like
people
people
do
do
small
small
cultivation experiments in the
experiments
lab,
in the lab,
and then
and then
they
they
publish
publish
paper,
paper,
and then
and
that's
then
all.
that's all.
So
So
they don't
they
move
don't
beyond
move
that.
beyond that.
But
But
we can
we
do
can do
more than
more
that
than that
to
to
transfer this
transfer this
knowledge
knowledge
to the
to the
community.
community.
So first
So
thing is
first thing is we can
transfer this knowledge to the farmers, where they can do the next level experiments, and then they can scale up the cultivation process, and then finally can go to the industrial level. So if you don't pass this scientific research to the community, I think we are doing a big mistake. So to go beyond, we need to pass our research into the community. So recently I saw one small mushroom farmer. He was able to cultivate our endemic Pleurotus giganteus, Kalupahana bimmal. So it's a very exciting news because until now we don't see our native mushrooms in our markets. So this is a very good news, and I hope he can scale this up, and in the future we can consume our own mushrooms in the market. So this is what we need to do as scientists, not just to research for papers, but do something beyond that.
Thank you, Professor Samantha. Professor Bhatt, how do you think an academic can maintain their scientific integrity while promoting innovation?
Yeah. I think I will explain this in two words. Integrity. What is required is honesty and hard work. I think this should be the force for any research and application to the society. If we maintain these two in our enormous honesty and hard work and that spirit of continuation. If this should be the driving force for any activity, especially some innovations. This is what our forefathers from the time memorial they said. Enormous integrity and hard work. Honesty. This should be the principle of any activity with us. If we maintain this and show in our work, I think we will achieve the goal. This is what philosophy of which I worked all along. We can also convince this to the policy makers who really take things to the logical conclusion at the end of the day. That's really what we should have. We should have to add. Probably we can ask.
Yeah, audience.
We have anything to add to this.
Yes, we will pass along the mic if anyone has any questions or suggestions. Or can also add to the speaker's ideas.
The thing is bridging knowledge, innovation and industry. So I think at college level or institute level, we should have a team wherein we approach the industry and get to know what are the problems in the industry. So based on the problem, then the institute faculty and the students can work on the problem, come out with a plausible solution and then hand it over. So that maybe because you're talking about funding as impediment, so in that case, the industry itself can fund that particular project and then the industry only will benefit, because then we make our students industry ready, and in that case the industry benefits, because when students go to the industry to work, they spend nearly 6 months training the students to work in the industry, and after they gain the experience, those people who work in the industry, they just leave that industry and go hop over to another. So that time they are left in doldrums. So for that not to happen, the industry and the institute should collaborate, make the students industry ready, so that when students go there to work, the industry does not have to spend so much on training them and then losing them in the long run. So that connect, if it is there, then that would be really very useful. And by doing this also, we are making our students very innovative, to think out of the box and get better ideas, because all these good ideas are from these young students if we give them a chance. And now with the present education policy, we have to inculcate into the minds that research is more than academics, because that is really very exciting. And if there is excitement, I think there is a lot of potential and production in whatever we are supposed to do for the betterment of our community. Thank you.
Thank you so much for that.
So my question is, now new generation always look at everything from this phone, right? So whether they will go for this stuff innovation in this microbiology application, because they want to earn very quick, right? So what do you think about this? How we can bridge the next generation gap, which is important?
Yeah, I need to very specifically answer that question. Yeah, this is something we have noticed. It's very difficult to get them that question. Yeah, this is something we have noticed. So we have started working with the smart devices. For instance, anyone who's gone abroad would know, and even if you go through your Facebook, you will see many of these scan type apps that come up. So especially, this was a discussion we took a long time ago, but we were being very careful not to put it into practice, because as a mushroom biologist, Professor Samantha would tell you, we can do these kind of apps where you can simply scan and that tells you what sort of a mushroom it is, but it can still go wrong. Sometime back, the two of us looked at the same photograph. I'm not a mushroom expert, and I'm more of a mushroom enthusiast, and we had a different argument. And so I was telling him, it doesn't look greenish to me, and then he was saying, no, it is greenish and this is poisonous. So that's what happens in certain aspects. So, but it's a good start, because I know when you go to other countries, you have these apps where they can use them to get information. That's one thing we can work on, but with mushrooms, we have to be extremely careful. And we are also using these mobile devices, because we have this collaboration with Michigan State. So they have developed what they call a multi-spec, which is a handheld device, which can basically scan and get the photosynthetic rate, and then based on the amount of data you capture, you can fit it into a mathematical model, and you can come up with various answers, like for instance, whether the plant is having any nutrient deficiency or this is a pest attack. These things can now be predicted. I think the younger generation like this, as you pointed out. So we need to capture that. And we have one more program with the ICT department and the industry linked to the cinnamon industry, where we are trying to work out, Nadisha and her team is working on this, so that they have the entire system from start point to end, and basically predicting what could happen at the next stage, and this is important for them. So sort of like a chain. So there are many different ways you can bring in the technology that they are interested in into this. It could be handheld devices, or it could be use of smartphones, or it could be developing apps, like even for farmers. I remember a long time ago there was an undergraduate project at the University of Peradeniya, which was trying to predict based on the scanned image what the disease is and what are the steps that could be taken, and even the progression is modeled. So these kind of applications, I think, would generate enough interest among the generation who are very much hooked on to the smart devices.
Thank you. Do we have more questions? Please feel free to direct your questions to our panel.
Yeah. I have a single question to both Professor Karunaratne and Professor Bhatt. My question is that how important do you think is regional collaboration in not just in mycology, in microbiology in general? Because as you pointed out in your presentation this morning, we are basically sitting on a treasure trove of bioresources in our region because it's very rich in biodiversity. I mean, Sri Lanka, the Western Ghats region, and even places in China. So how important do you think is for us to collaborate, and how best we can collaborate between countries in our region? That's my question to you both.
I think the collaborations are definitely possible and most regional collaborations, especially in the Asian region. And there are guidelines, national level guidelines, and within the framework of this, and if we develop the basic understanding between the institutions, and this can be taken further to the government level. And there are empty number of examples we have, collaborations between the countries. And certainly possible. And it is also important because this kind of collaborations will be helpful to the regional development. And this is, for example, we have done this at our individual levels, what we have in collaboration at the moment. But this can certainly be extended to at the government level and for a long duration exercises. Definitely possible. And we should take this initiative during the course of time. Yes. We have much, but we should do this.
So, as per your question, the collaboration. So sometimes in Sri Lanka, it's a big problem that scientists face, like there are not enough facilities. So we have such a high diversity of mushrooms and fungi, but the problem is we don't have good microscopes, we don't have sequencing facilities. And in such case, the best way is to collaborate with other institutes or universities in the regional, like China or India. So in this way, we can move forward. Not only saying that we have a high diversity of mushrooms and fungi, but if you don't like survey them and find out what we have, then maybe without knowing, some fungi or mushrooms extinct. We don't know because we don't know what we have. So that's why we need to collaborate with other institutes or universities. And we can do this like in the best way, like signing MOU or MOA, mutual understanding or mutual agreement. So this way we can discuss how we do our research in collaboration with other institutes or universities. So you can see most of the good research people are doing not by doing their own, but by collaborating with other institutes or universities. Yeah, thank you.
Very nice session. I had a talk with Professor Samantha, and this is in regard to indigenous mushroom varieties like we have in Goa. We call them ulmi, and they're very prized. And recently we had an issue wherein most of the families, it's like there are very few people who know to collect them, where to go to collect them, and they're highly prized for that reason. There was a case in Goa recently that by mistake some poisonous ones were collected and then there were issues, they're hospitalized. So how do we tap that? And usually those people don't like to give on their resources, they like to give it to their own generation. But the generation which they are in, they don't like to take it in their own family members. So how do we tap that? They are the ones who are locally been used, they know how to do. How does it come to the industry level in this, how the gap is bridged? I don't know this. How can we tap that?
Yes, this is the problem of many places, not only India. I think even in China, in Yunnan, we experience like when people know where these prize mushrooms grow, they don't like to share this information to others because they are afraid that others will come and harvest them. So, but it is very important that this traditional knowledge pass through generations to generations. So, in China also, when we go to village areas, first thing we do is we find some locals who know about mushrooms, and we talk to them and see what they know about local mushrooms and what they know about edibles and poisonous mushrooms. And then we try to understand what is the scientific the facts behind this traditional knowledge. So, for example, in Sri Lanka, people believe that if the mushrooms are very colorful, they are poisonous. And if a mushroom has a ring or vulva, they are poisonous. But these are not true. Like for some specific mushrooms, these are true, but not for all the mushrooms. So the best thing is we talk to the locals and try to grab some knowledge that they have, and then we try to like link these to scientific knowledge and pass to generations to generations. Otherwise, they disappear without knowing. Without. Yeah, so it is very important to document these things and pass to the other generations. Yeah. Thank you.
Thank you very much, Dr. Personali and the team for discussing this very valuable topic. Actually, I have a suggestion. Basically, I am an archaeologist. I also having the same experience. Like for the basic things, it's very difficult to find funds, especially the basic sciences. Therefore, we have to change our mind to deviate, especially focus to the applications of lichenology and so on. So as Professor Samantha mentioned, now we have to have some research related to the basic sciences. Now as we are discussing now, sometimes we have to focus to identify many of the species. But in case if you apply for the funding, it's very hard to find funds in that aspect. So at least if we can convey this message to the government or other organizations who are funding, at least one portion of the particular amount for the basic sciences, because now in the case of lichens, now we have at least we know about 10% of the total lichens in Sri Lanka. So about 80% is to be discovered. So in that sense, because of the limitations of the funding, sometimes, as we discussed, some species may vanish. We don't know what would happen or what has happened them to the past. So this is my suggestions that if some possibilities are there, we better to encourage government and other funding organizations also to make some decisions. At least particular percentage need to be donated for the basics research. Thank you.
We have time for one more question.
Okay, thank you very much for your nice panel discussion. I am from the Forest District State Timber Corporation. We have some experience in some wood destroying agents and some agents affected to the forest plantations, especially eucalyptus species in the upcountry area. So I would like to know some other country experiences, especially from I like to ask from Professor Bhatt. India or other tropical countries can get some experiences, some research, ongoing researches on this field for identification of this type of disease or lichens or any other species for an remedial actions. Thank you.
Honestly, I don't know much about that, but I will find out these informations and pass on in the course of time. Because I don't know whether Ashish, you have any information on that?
Present, we can get connected to some people who are working in this field here. Pathology, plant pathology. Yeah, we need to find. Find out the causal organism, and we can connect to the expert in that particular.
Dr. Mubala, thank you for your question. We have in China, they have eucalyptus research center. Actually, it's huge, and they have a lot of. They are collaborating with different universities, especially for agroforestry. I know it very well, and I know some people. Maybe I think we have discussed about this previously, and I should say when we are talking about the comfort zone and planning research, Dr. Muthumalai from State Timber Corporation is one of the best resource person we have ever had at the moment to working with us. So when we planning research, so of course we should have one person from the government. Of course, he's a policy making level, and he knows what would be the outcome or expected outcome from the particular research. So it's very important to have that type of person in your research group. So we know that we are working with one student in the University of Colombo. So he was always, okay, we should do like this, we should like this. So yeah, that his perspective is from the applied level. I mean, sometimes we are as a fundamentalist, we cannot see some aspects, but from the industry or maybe policy making or government sector level, he can give us very good suggestions. So I think maybe as Dr. Dialect also told that we should have, I mean, the students with good mastermind, of course, with very creative ideas. And some, I know Professor Samantha and Professor Pai very well, they're always very humble to students' ideas. So we also try our best to listen to them. And as you told, Professor, that in the morning we have our students, especially particularly Madara. So she came up with all the ideas by herself, and she discussed with Professor Chaturanga. Okay, it's yours research, you plan it. That's called networking. And fortunately, they have material science department, and some people from the material science department, they have very good listeners, of course. because we are not material science people, so we have to tell our ideas to them. Then they should be humble to listen to our ideas, and then they can work on it. So it's very important, and still I feel it's in the comfort zone.
Thank you so much, everyone, for those engaging questions and thoughtful answers. So, to conclude, I'd like to hear from each one of you a key message for the next generation of microbiologists. Let's start from Professor Saman.
As I mentioned earlier also, let's not just focus on the publications, go beyond the publication and do something to the community. So, as I mentioned, taxonomy is very important because we need to find out what we have, and then we also can make the action plans for conservation. But not only that, go beyond that and do some value addition to the species that we describe, and then pass it to the community. And for example, the mushrooms, there are a lot of like medicinal or edible mushrooms in Sri Lanka, but we still don't see a lot of mushroom-based products in our country. But if you see China, the biggest mushroom producer in the world, so if you go to a market, you can see like more than 800 different types of edible mushrooms, edible and medicinal mushrooms. So in Sri Lanka also we have several different edible and medicinal mushrooms, but because of the lack of knowledge, people are afraid to eat, afraid to consume. But if you do proper research and pass this to the community, then people understand what are edibles and what mushrooms can be used as medicines. So this is what we should do as microbiologists or taxonomists. So I hope the young generation, they think out of the box and do some applied research in the future. Thank you.
Yeah, thank you very much. Yeah, what kind of message? I have seen the last, I'm just giving a, overviewing the last 5 decades, what happened, especially from some message from India, what the experiences what we have. The taxonomies really worked as a separate entity in the 1950s, 60s, 70s, those times. And each one was a giant in their own areas, whether it is in plant taxonomy or fungal, animals, all these areas, different areas, people worked independently, number one, especially the taxonomies. Physiologists work separately in the academic sector. The application was not really focused much. But the time changed at the turn of the century, and the policy makers at the government level started to ask questions. Why do you need funds only to study the taxonomy? That's enough. You should come out and see where it is applied once. So suddenly the focus started to change. Everyone started to become alert, and where the application, how to bridge with this. Because industries also were not really looking at the academics, what they will give. They were working from some of the protocols which we learned from abroad. And we wouldn't see much diversity in the industrial level also. But things kept changing dramatically in the last 20 years. Those people sitting at the top, at the government level, started to ask, what is your contribution to society? How this will help us? So everyone started to become, and we also have certain sectors in the Council of Scientific Industrial Research, which looked for really the application of science. So people became alert and started to bring the linkages. And in the last 10 to 15 years, really there is a big push for this kind of stuff. So the generation next will definitely have enormous opportunity to take this science to the society and really to make an impact. So we look forward that the basic science in the meantime has put up a very strong foundation, and it is giving a lot of opportunity for application. So that will be the focus in future. Even our educational system, the University Grants Commission has come out with policies that even the syllabus also has been tuned, that everything what we do should be applicable. And each of these government is pushing to bring up startups, newer startups. About 10 years ago, we had a few hundreds. Now it's in thousands, the startups, and so much of money is pushed to. Many of these startups have never taken from the ground, but few have really made an impact. So there is enormous opportunity for the basic science and its application in the coming days. So I look for a lot of sparks in the generation next, in the coming times. That's the optimism we have. Yeah. Thank you.
I believe I've seen several generations. So for the next generation of microbiologists, it's important to have information at your fingertips, because one thing is people are moving away from traditional reading. So if they enjoy other kinds of reading, so be it, but you still need to identify the correct sources and be ahead of the game. That's the modern aspect of it. And the next one is be humble enough, as Professor Nalin pointed out, to listen to people, because strange things may happen when you speak to especially people with traditional knowledge. You might pick up on certain things that later on you might work on. But in order for you to work on, you need to have the information at your fingertips. Then you see your way out of it. And the other aspect is collaboration, of course, because many heads are better than one. We may see a problem in a single perspective, but if you have a group, the group is capable of coming out with the best solution. That's what we are doing at the moment. We have several people coming from different disciplines working with us, and sometimes they propose things we didn't even think about at the beginning. So with the next generation of microbiologists, in order to make an impact, you need to have information at your fingertips. You need to collaborate with people, and you need to be open about it. And there's no holier than thou attitude, because if you have it, you're the one who will lose. Information can even come from the village coffee shop. You never know. So that's the message I want to have.
It's a nice question, I should say. And finally, I believe the creativity is the number one always, because they have to think out of the box, as some of our experts told. And of course, we have to be very humble, creative, and listen to others' opinions, ideas. And we all know that now we are in the age of using AI seriously. So we have to be very careful. So I'm not against AI, but the thing is we have to use it very carefully. So in the future, they're saying that the people who work with AI might replace the people who are not work with AI. So that might be happening soon. So we have to be very careful, and we have to work it very in good way. So of course, if you are a creative person, I think you don't have to worry with AI. So that's one of my opinions and suggestions and as advices. And of course, you have to work with other people, not particularly in your discipline. So I always believe multidisciplinary science. So it's very important to have more opinions, ideas, and of course, look into your research in different aspects. So maybe multidisciplinary sciences will also be very important for new generation scientists. Yeah, thank you.
Perfect.
So on behalf of the organization committee, I would like to extend a heartfelt gratitude to all of you for sharing your insightful ideas with all of us. With that, we come to a conclusion of our roundtable discussion today. Thank you so much.
Ladies and gentlemen, we have a few announcements. Please proceed for lunch at Bite Cafe at the ground floor. And after the lunch, kindly join 3 parallel symposium sessions taking place on the 5th floor of Genesis building. Please note that session number 1 on agricultural, environmental and forest microbiology will be held at room number 502. Session 2 on microbial diversity and taxonomy will be held at room number 501. Session 3 on microbial technology and industrial applications will be held at room number 503. And transportation has been arranged outside the auditorium for the session. So thank you all for joining. Have a nice day.
That's really good. In application will be found later. किधर स्पेशल है किधर आंसर दे आंसर प्रोडक्शन देखिए चढ़ने वाला प्रोडक्शन देखिए चढ़ने वाला आंसर
This at 1:30 p.m.